Okay, good evening everyone and welcome to week 4 of plant physiology. And over the last 2 weeks, hopefully you've had a chance to look at lectures 4, 5 and 6. So here's lecture 4, the xylem. Lecture 5 was stomata, the lung of the plant, and lecture 6 was transpiration. So tonight I'd like to revise the most important concepts, the major concepts from these 3 lectures. But while I was waiting for students to join the class tonight, I noticed this amazing image here. This photo here, and obviously you can see that these images are of xylem elements. But look at this here, this coiled structure associated with the xylem. Does anyone know what this is? This coil. Oh, you don't have any audio, Luke? Okay, Lachlan and Amelia, you can hear me okay still? All good, okay. I'll just let Luke know. Okay, something wrong with his internet there. All right, so can anyone imagine what this coiled structure is that's associated with a xylem vessel element? And think in terms of that reinforcement, that strengthening of the xylem that makes it able to withstand those enormous pressures. Those enormous negative pressures. Any idea what that might be? And if you've got no idea, feel free to just say, no idea. Or you can say it's a ghost. I liked that answer. No? All right, who remembers that we talked about the strengthening of xylem by lignification, by the formation of lignin? That's what we're actually looking at here. These coils that form are made from lignin, and they are incredible structures, incredibly tough structures. That's what you see. If you can dissect plant tissue under the microscope, then occasionally you actually pull out one of these coils. You'll remove the coil from the actual xylem vessel element. Quite amazing to actually see it under the microscope when that happens. Yeah, it's amazing, incredible. I have wasted much time looking at these coils under the microscope because they're just quite amazing to see. All right, so I'll just move through this. Yeah, I'm very good at procrastinating, wasting time. All right, so we've talked, we've looked at the xylem structure. So the structure and the function, so xylem vessel elements and tracheids and pits. So remember the pits and pit pairs? They're basically almost like pores on the side of the element walls or on the side of the tracheid walls, and they seem to pair up with a pit in the neighbouring vessel element. So pits or pit pairs, and I don't want to go over that because that's very straightforward. Wall xylem structure, vessel elements, tracheids, and of course those interfascicular, what they call interfascicular fibres, which are very also cells involved in structural support of the vascular tissue. Some nice images here of tracheids, and here are the pits, pit from one tracheid, and then that's aligning with the pit in the neighbouring tracheid. Some more nice images and some images again, and once again. All right, here are some more pits, and I will get there in just a moment. Here we go. All right, so we're already familiar with the concept that water moves under a large tension, a large hydrostatic tension through the xylem. We've talked about that last year and also in this subject. We've also mentioned the problems that can be associated with this tension. So firstly, that sort of tension, that negative pressure in the xylem imposes this inward force on the walls of the xylem. And so that just looks like this. So if they're the walls of a xylem vessel element, then that negative pressure or tension causes the walls to curve inwards, because you can imagine that tension, of course, is like a suction pressure, suction force, I should say. And so what is it that prevents those walls from just caving inwards? What prevents them from caving inwards? Because plants, of course, can deal with this very well. And we were just talking about it earlier. Structural components, yes, of the xylem. What are those structural components? What is it? Lignin, yes. So thickening of the secondary cell wall and lignification. Quite amazing stuff. Cell wall lignin, good. All right, so we know that plants can deal very well with this problem here. That's not a problem at all. And I'm not sure if I've actually described this to you before, but I will bore you with it. Well, I don't think it's boring. Bore you with it once again. I was looking at lignin years ago, doing some work with lignin, and this involved, and I'm drawing this definitely not to scale. So I've just drawn a microscope slide, and on that slide, I had a very thin, tiny section of plant tissue, and I'll just draw a few cells in there. So this is supposed to be a three-dimensional representation of a transverse section. Anyway, as part of this, the chemical reaction that I was actually carrying out to show the lignification, I was looking at presence and of lignin and the decrease in lignin as a result of knocking out certain genes that we thought may be involved in lignification. So as part of that, I had to use a little bit of very concentrated hydrochloric acid and just add it just for a few seconds and then immediately wash it off. Now, if I was a bit too slow, which is quite possible, this is what would happen. So here we have, what I'm trying to draw now is just the xylem vessel elements. So we've got them in bundles here. Okay, they would remain like little upright columns on the slide. So that's what I'm trying to draw here. They would remain upright, and the rest of the cells would basically just disappear. They would just turn to mush because of that, leaving the acid on there for too long. So this just shows, I think it proves once and for all how tough that lignin, that substance lignin is. Even with concentrated HCl, they just stayed upright, and the other cells virtually dissolved into nothing on the slide. And all I had was basically a mess around those little columns, and I had to start again. So there you go, more time wasting from me. And I'll just clear that. All right, another problem that we can see here. Okay, water under pressure, well, under the type of pressure that we're looking at here, is physically unstable. But if anyone's done any further reading, we can actually extend this a little bit. And the water that you find in the xylem in a plant is actually said to be in a metastable state. Metastable. Anyone know what's meant by metastability? I'll just put that word in there for you. Water under that amount of pressure is said to be in a metastable state. Anyone familiar with that? Metastability. Big? Or sort of, not quite, not quite. Good try. Water under pressure in the xylem is actually in a metastable state. Ideal. Oh, it's actually the opposite. Well, not quite the opposite, but it's not quite the ideal state. Above, beyond stability. All right, so you're all sort of, you know, going around. Sounds positive. Sounds positive. All right. Now, I think this is really interesting. It's bad. Well, actually, it's not bad. It's not what you would normally expect. I think that's the best way. Not what equilibrium. It's actually not what you would normally expect given the current situation, if you like. But I found, let me just bring this up. I found this, which I think illustrates the concept of metastability really, really well. So firstly, let me give you a little bit of a definition. So when something is in a metastable state, it's existing in a stable state that is not its lowest energy level, if that makes sense. It's existing in a stable state that it would not normally exist in, because as we know from the laws of thermodynamics, everything proceeds from high energy to low energy. Everything proceeds towards entropy. You'll remember that, yeah? And that's how water apparently exists in the xylem, in a metastable state. So it's not at its lowest energy state. It's actually existing as a liquid when it actually shouldn't exist as a liquid, given the enormous pressures that it's under. And I think this illustration is really nice here. So what it's showing here is a ball it's just sitting in this hollow, and it's quite stable sitting there. It's not its lowest energy level. Think about rolling a ball down the hill. The lowest energy level would be at the bottom of the hill, wouldn't it? Okay, so you can give this ball a little bit of a push and it'll rock backwards and forwards and it'll still stay in this hollow. In order for that ball to get down to its lowest energy level, it has to overcome this energy barrier. You've got to push it enough so that it goes over the edge. So I thought that was a nice illustration. So to try and compare this or relate this to water in the xylem, the water in the xylem, the water in its lowest energy state would actually exist as water vapor or the gas phase because of the pressure that it's under. So we know that it's easier to vaporize water if you increase the temperature. Yep, that's pretty straightforward. But water can be vaporized very readily if you put it under enough pressure. Yep, same sort of thing. But the water remains liquid. And this occurs for a couple of reasons. One, because of the properties of water, because of the cohesion and the adhesion properties of water. Yeah. So this basically makes this barrier, this free energy barrier, for transition from the liquid to the vapor phase very, very high. It increases this energy barrier. Also, another reason is because of the structure of the xylem. The structure of the xylem is very, very good at preventing sites of water vapor, sites of what they call air seeding sites, where bubbles will form, and we'll talk about a bit more about that process in just a minute. So I think that's very, very interesting. very interesting concepts to understand, at least in a little bit of detail, why that water is said to be in a meta-stable state within the xylem. Isn't it amazing? Isn't it amazing how much chemistry and physics underlies biological processes? Isn't that amazing? Confusing. Yes, definitely confusing when you first hear about it or you first read about it, but fascinating nevertheless. Okay, so water under pressure is physically, is in a metastable state. So what happens? What can happen to that water column in the xylem? Of course, bubbles can form. Yep. And who remembers? Yes, cavitation. Yep. Fantastic. Good. Thank you. Yes, absolutely. Water has a lower boiling point under pressure. Absolutely. Absolutely. And this is really nicely illustrated. If you place a little tube of water into a vacuum chamber, and then, of course, close the door, evacuate the chamber, and you're basically, the pressure's going up to about, you increase the pressure to about 15 psi from memory, which is roughly equivalent to 1 atmosphere. If you do it too fast, you'll actually vaporise all the water in the tube. And we used to do this to try and get the air bubbles out of the water. The air bubbles would be trapped with the plant tissue in the actual, it wasn't just water, it was a chemical solution. But you had to be very careful as you're increasing that pressure. So yeah, absolutely. But anyway, back to cavitation. Cavitation, why does cavitation occur? So in your reading, did you see any information about why air bubbles can form in the xylem? And we know that the formation of air bubbles actually disrupts that flow of water, the movement of water through the xylem. But let's first start with why bubbles form in the first place. What could actually cause them to form in the xylem? Cavitation. Formation of air bubbles that break the continuity of water transport. Why can bubbles form? Slow speed? Slow speed, do you mean slow speed of water transport? Water movement? Look, that's possible. That's possible. I'm not too sure about the slow speed. I'm more familiar with water moving fast, but in the presence of a water deficit in the soil. When there's a water deficit and transpiration rates are high, therefore you get very rapid movement of water. Unstable under pressure, yes, but keep in mind that water movement, if there's no problems, which we're going to dissect in a minute. If there's no problems, if there's abundant water in the soil and there's no other problems, then we don't have a problem with water under tension. There's no problem with water under tension. The gas bubbles don't actually form. All right, let's think about now water entering the roots. And when water enter the roots, to get into the xylem, the water actually enters through those pits, through those pit pairs, to get into the side of the xylem vessel. That's how water first enters the xylem. Now, those pits, or those pit pairs, or let's just say the pits, those little pores, they have a very, very good sieving mechanism. They generally don't allow air bubbles to enter the xylem. But imagine there is a great deficit of water. The water's still moving in, but you'll get that sort of bubbling or effervescent effect as the water's drawing water in behind it, but it's also drawing in air because you've got these air pockets that are forming in the soil solution as the water's drying out in the soil. So air bubbles can be introduced in that way in cases of severe soil water deficit. In particular when that pressure difference across the pit, the pressure difference is great enough to overcome the capillary force that's occurring in the actual xylem itself. And this is called air seeding, the introduction of an air bubble into the actual xylem, air seeding. All right, now there's another interesting way. Before I come to that one, imagine also that there's some sort of wound to the roots, or some sort of wound anywhere within the plant, and that wound may extend through to the xylem, and that's another way that air can enter. Yeah, through some sort of wound. It might be insect damage. It could be disease through fungi or bacteria that are enabling that sort of exposure to the xylem to the air. So that's another way, but there's also another very interesting one. And they've done, you'll be interested actually, all three of you in this one, because they've done some of this work in grapevines. So imagine this, imagine what happens if the xylem sap freezes. And they've shown this in grapevines, if the xylem sap freezes. Do you think that dissolved gases are very soluble in ice? Dissolved gases, they're not, exactly right. Dissolved gases are not soluble, not very soluble in ice. So freezing of the xylem sap, the xylem water, can actually lead to bubble formation, so that when thawing takes place, you have all these bubbles forming in the xylem sap. Okay, so that's how cavitation, absolutely, frost damage, absolutely, yes. And I should have actually included the paper that some work was done on grapevines, but it doesn't matter, I've got another interesting one for you that's also in grapevines. Okay, so when the bubble is formed and it starts to expand, which it can do, that's called cavitation, which we know. That gas-filled space that starts to occupy the xylem vessel is known as an embolism. And you'll all be familiar with this term. Where else do we hear about that term, embolism? Where does that embolisms commonly occur? Yes, yep, occur in the brain and also other parts of the blood circulatory system. Medical shows, yeah. What about deep sea divers? That's another another time when you commonly hear about that. So embolism forms, yes, yep, absolutely. Embolism forms, but the size of this bubble actually prevents it escaping from an individual vessel element. So how do plants actually overcome, the formation of a gas bubble? How do they overcome it? And while you're thinking about that, stomata. Not sure about that one, Luke. Don't know about that one. Yeah, no, that's all right. Any idea how plants can overcome, which of course they can overcome, and it's a very common occurrence, formation of embolisms. But of course, because plants are so amazing, through the leaves, shut down that tube. Yes, now that's one. That's actually one mechanism that has been shown that they actually shut down that particular vessel element. And I'll just go back to here. They shut down that particular vessel element, and the water, can you see these arrows down here? The water simply detours through those pits into neighbouring vessel elements. So that's actually been demonstrated. Absolutely. Okay, and to answer that in a little bit more detail, let's just look very briefly at a couple of the results obtained here. So this paper, published in New Phytologist, water uptake can occur through woody portions of roots, and this facilitates localised embolism repair in grapevine. So amazing. Most people believe that the woody portions of roots, that is the older portions of the roots, there is no water uptake and no solute uptake through those portions. But that's actually not the case. So it's amazing what you believe that's actually been in the literature for many, many, many years. Oh. And then research will come out and show that that's not actually the case. All right, so I'll just show you a couple of figures here. So I think here they're looking at roots. Let me just have a look here. Yes, woody roots, grapevine woody roots here. So this is just a section here, a section of a woody root, or the outer part of the woody root. And this is at 0.2 hours after the first image. Okay, so in yellow here, this represents, the yellow represents basically areas of the root that lack water. Let me just make sure I'm reading this correctly. In yellow, don't want to tell you the, yes, so airfield volume. So where you see yellow, this is air. So you can say that there's a lot of embolisms here in this section of the woody portion of the root. Then after it was watered, the actual root was, that portion of the root was watered, the woody portion. 12 hours later, look at the reduction in air. So now we've only got a small region of that woody root that actually contains air. In other words, water can penetrate that suberized woody portion of the root and repair the embolisms that have formed. So I think that's very interesting. Let's move on to one more figure here. And we're looking at, no, it wasn't that one. Here we go. So now I think they're looking at the actual, still woody roots, grapevine woody roots. So this is at 0 hours, this one here. And this represents embolism here, this dark region here in the xylem. 12 hours after watering, you can actually see that it's resolving. That cavitation is actually resolving. And then 20 hours afterwards, it's almost resolved. So we almost have restoration of water transport right through that xylem. So pretty amazing. Oh, we've got a question. I thought that was amazing. Okay, the question, or maybe it's just a statement. When layering grapevines, okay, you have to help me out here. Layering grapevines, what does that mean when you're layering them? Water is taken in through the woody bark to form roots. I'm not sure if this is similar. Layering. Are you talking about air layering? Is that what you're referring to? When you're propagating by air layering? All right, we'll see if... Okay, so cavitation. We've looked at cavitation and also, oh, here we go. No, burying old cales to form a sister vine. Canes, that I'm sure you mean canes there. Yeah, thank you. I was about to ask what cales were, but yeah, thank you. Burying old canes to form a sister vine. Okay, so hardwood cuttings basically for purpose of propagation. Yeah, okay, absolutely. Well, yes, it could be, except that when you take hardwood cuttings, obviously you've got an open end. But yeah, look, obviously there is water can move through that woody tissue and basically penetrate the xylem in that way to alleviate the effects of cavitation. So quite interesting in that way. Now also there is another way. So when transpiration slows down or ceases altogether, that the pressure on the xylem sap is alleviated, so less pressure. And when that happens, then those gases within the air bubble can actually be dissolved once again into the xylem sap. So that's also another way that cavitation can be relieved. And one more way. We haven't talked about root pressure in much detail, but root pressure is thought to be another way that plants can restore xylem function. All right, enough on cavitation. As interesting as it is. All right, so I think that's enough there. Yeah. All right, we'll leave that there and move on to, so this was lecture 5, stomata. Okay, this is the slide that I was after. All right, now we're going to look at more detail in at water movement. You're all familiar with the concept of the transpirational pull on xylem sap, and we know that this is transmitted all the way from the leaves to the root tips. Of course, only when transpiration rates are really high, then that negative tension can be transmitted all the way down to the actual roots. And we also know to a certain extent that transpirational pull is facilitated by certain properties of water. Okay, so let's start by looking at this arrow here. And we know that water moves from the soil solution into the plant and up the plant along a water potential gradient. So I want you to tell me, based on what you know so far, is that movement from a high water potential in the soil up to a low? Sorry, does it move from high water potential here to low water potential, or is it in reverse? Should that say from the roots? Should that say from the roots? Oh, hold on. From the roots to the leaves. No, that's actually correct. The transpiration, and I'll talk about that why in a minute, because we're going to look at how that transpirational pull, in other words, how that negative pressure, how it's initiated in the actual leaves themselves. So the basis of that negative pressure in the leaves. All right, so does anyone remember water potential gradient? Does water move from high to low or in the other direction? Pressure is very low in the leaves. Yes, correct. Thank you. So high water potential here, down here, and very, very low water potential up there. Okay, good, because that's always a source of confusion, and we'll cover that in a little bit more detail in just a minute. So I really do like this image because it shows basically, well, it doesn't actually explain it, but it's showing that there are processes that are occurring here in the actual soil solution that contribute to water movement and therefore water uptake. Then we have water moving up the xylem, and we can look at those properties of water that actually contribute to this. And then, of course, there are processes occurring in the leaves themselves, which also contribute to movement through the xylem. So I'd like to actually dissect each of those in turn. We've already spoken about all three of these processes, but in a very fundamental way. So I'm quite sure that you guys can extend on what you already know about these processes. Now, I'd just like to go a little bit further just to show you something, which I think is in this slide, in this lecture rather. No, maybe not. All right, let's go back. All right, so just hold that thought for a sec. All right, so based on what a couple of you have just said, we go from high water potential, water moves from a region of high water potential to a region of low water potential. What do you think about the labels here? Here and here. Are they right or wrong? This is a very good test. Wrong. They're actually wrong. Absolutely. Did anyone pick that? Did anyone look through these lectures and go, hang on, something's not right there? They're actually wrong. So, as some of you said, we're talking about movement from a region of high water potential. High water potential means that the value is approaching zero. That means it's less negative, not more negative. Yeah. Low water potential, which would be up here in the atmosphere, for example, low water potential means that it's more negative, a more negative number than what we have down here. Yeah? So an example, give me a number, just any number that may approximate water potential in the soil. What do you think? Any number, doesn't have to be that precise. Just to illustrate the concept a little bit further. Okay, beautiful. Negative 0.1. Let's just say the water potential of the soil solution is negative 0.1. Great. Now, I don't know if everyone else can see that, so I'm just going to type that in as well. Thank you. Soil water potential. Okay, there we go. Thank you. So what do you think would be just a very rough figure for up here in the atmosphere? So a value for the water potential. -1. Yeah, all right. We can say that. It's actually even great, even more negative than that. Even more negative than that. So anyway, the point here is that water potential is higher here. That is, it's less negative. It's still negative, but it's approaching zero. Okay. And it's a very negative number here. Very negative. It may be in the order of negative 100. Negative 100 in the atmosphere. Okay. Yeah, absolutely. Yep, negative 200. Well, that's that's really, really low, but yeah, but you can see what I mean. You can see that enormous difference in water potentials from high water potential down here. Yep. Yeah, maybe a bit too much. To very, very low, in other words, a very negative number in the atmosphere. Okay, good. That's terrific. All right, now I'd like to spend a little bit of time just looking at what happens here. Why is the water potential, the values for the water potential down here, why are they the way they are? What contributes to those values? And so let's have a look firstly at soil types. So just very, very broadly, 2 soil types. Obviously, there are a lot more soil types. So we've just got the 2 extremes here. So water content and the rate of movement in soils obviously depends on soil type and structure. Sandy soils have large particles and therefore they have a low surface area per gram of soil with large spaces. Clay soils, on the other hand, have very tiny particles, 2 microns, for example, in diameter. Therefore, clay soils have a greater surface area per gram of soil and much smaller spaces in between the smaller particles. Of course, after watering, water moves down through spaces, either displacing or trapping air. The process of capillary action, capillarity, results in the smaller channels being filled first. And of course, there's a lot more of those smaller channels in a clay soil. And of course, based on the properties of water that we're already familiar with, water can adhere to soil particles as well as filling those small channels. All right, so let's now talk about water potential of soil water and what contributes to those values that we're familiar with. All right, let's start with what makes up water potential. And you remember, question, okay, more with toiletries, or is it more about leaf area? Okay, so are you referring to the actual water potential? Water potential of the atmosphere, the higher negative pressure. You mean the higher negative pressure? Oh, right, the atmospheric pressure, you mean? No, that doesn't have anything to do with the actual with the height of the tree itself. No. Okay, now I know what you mean. No, I don't know. Yeah, I'm not sure about the negative 200. That was just a bit too much. But yeah, just more around negative 100 or less than that. Yeah, in terms of the water potential of the air. All right, so let's dissect water potential. What makes up water potential? What contributes to water potential? Who remembers? 2 major contributors to what we call water potential. What are the 2 major contributors? And they're actually both mentioned here on this slide. So remember, water potential equals, yes, solute potential is one of those contributors. Solute or osmotic potential, correct? And what's the other contributor to what we call water potential? Pressure, correct, yes. So you'll remember that water potential equals, yep, solute potential plus pressure potential. And there's actually a third one, which is not normally included. Anyone remember what that one was? It's not normally included by plant physiologists. Gravity. Gravity. Tidal pressure. Tidal pressure. Oh, I don't know about that one. Not sure about that, about tidal pressure. All right, so let's look at solute potential. So the solute potential of soils. And this might surprise you that it's generally negligible. And that's because the solute concentration is normally quite, solutes in soils are normally quite dilute. They're found in very dilute concentrations. So generally the solute potential of a soil, except for saline soils, is only in the order of about negative 0.02 megapascals. Very surprising. All right, the second part of our water potential is pressure. In wet soils, so if you have saturated soils, the pressure potential is 0. But as the soil water starts to dissipate, as the plants are using it, or it's evaporating, or it's simply draining down, then the pressure starts to develop. And that pressure is a negative hydrostatic pressure. Who would have thought? Where does this negative pressure actually arise from in the soil? Where does the negative pressure arise from? Let's take a look at that. I'll just get rid of that. Negative pressure in the soil. All right, let's read a little bit here. Water has high surface tension. We know that. Water has very high surface tension. And this surface tension will minimize the air-water interface because of that surface tension. However, due to the adhesive properties of water, water will also cling to the surfaces of the soil particles. Let's try and visualize that. And I'll just get the pen back again, looking at this great image here. So we have a particle, soil particle here, well, sand, clay particles. In blue, we have water, in dark blue and light blue. In white, we have air. So air pockets forming as the soil starts to dry out. All right, when we talk about air-water interfaces, look at what happens because of surface tension. Because of surface tension, you end up with these curved air-water surfaces. And you can see that here. As the water recedes into these channels here, into the spaces between the particles, because the water, of course, clings, it adheres to soil particles. As the water recedes because of surface tension and the adhesion properties, we end up with these curved air-water interfaces. What does that mean? Water under a curved surface develops a negative pressure. There it is. Such an important statement here. I should have put that in big bold letters. Water under a curved surface develops a negative pressure, and it's amazing how great this negative pressure is depending on the radius of that curvature. And we'll look at that in just a moment. So as the soil starts to dry out, the pressure becomes even more negative due to increased curvature of the air-water interfaces. Is everyone still with me? Kind of. Come on, I know you guys can follow this. I know you can. Physically here. Amelia, you need another glass of wine, maybe. Or did you already have one? All right, and don't worry too much if you don't fully understand this. No alcohol tonight. No, no, you don't have to fully understand this, but be aware of it. Be aware of what's actually occurring in the actual soil and basically the conditions that are driving movement of water. Why can't I get rid of that? All right, so, okay, water will move primarily in the soil as a result of bulk flow. And you're familiar with that term. Bulk flow refers to the movement of molecules en masse in response to a pressure gradient. So we've just seen how the pressure develops as the soil water recedes. And now we're looking at how water will move from one part of the soil to the other part. So for example, as the soil solution around the roots actually gets all used up, that soil water gets used up, then how does water move from another part of the soil towards the actual roots of the plant, which is what we want, obviously. So we've seen that pressure in soils is a negative pressure, and this develops due to those curved air-water interfaces. We know that water will flow from high soil water content to low soil water content. In other words, it flows towards those smaller water-filled pores that have the more curved air-water interfaces, in other words, the higher pressure. So as plants deplete soil of water around the roots, the pressure reduces and establishes a pressure gradient. So that means water moves through the soil as a result of bulk flow and along a pressure gradient. That's all you really need to take home from this. And if you wanted to know about it in more detail, then you know that there's a bit more involved in terms of setting up that pressure gradient in the soil. All right, what about the rate of flow? The rate of water flow in the soil depends on two factors: the size of the pressure gradient and the hydraulic conductivity of the soil, which simply refers to the ease of water movement through the soil. And you can imagine that water moves easier through sandy soils, so they are said to have a larger hydraulic conductivity than clay. Okay, water absorption by roots. I won't talk about this too much. We've covered this exceptionally. So the three pathways of water flow into the root, you know all about the apoplast, symplast and transmembrane pathway, and you know how important it is to know this. And the Casparian strip. All right, let's now look at root pressure. Does anyone need a quick break before we go on? Quick break? I could handle a 5-minute break. What about you guys? Oh, I think you're already gone. You've already left me. You know, a student told me the other day that they fell asleep on slide 24. I said, I don't want to know that. I don't need to know. Now I'm going to think that all my online classes are boring. Yes, 5-minute break. I'll see you in 5 minutes. That's why the camera's off. Thanks, Amelia. All right, see you in 5. Who got stung with that first lecture? What does that mean? Who got stung? That first lecture. Okay, let me know when you're ready. Let me know. Back. Okay, Amelia, are you back? Yes, excellent. I don't understand that question, Lachlan. Who got stung with the first lecture? Waiting for the first lecture to start. The camera was still on. Oh, yes. Yes, I heard. I didn't see that, but I did hear about it. Yes. Oh, that's okay. They must have been enjoying themselves. Terrific. Okay, let's move on. A little bit about root pressure. So you might remember that root pressure is a positive hydrostatic pressure, and plants tend to move water through the xylem only using positive root pressure when transpiration is very low or completely absent, of course, if the stomata are closed. And there's a really neat way that we used to demonstrate this years ago, and I think we should do it again. We would take a large tomato plant and we would cut the entire shoot off. So we just had just a little part of the stem remaining. The soil was very well watered, and then we'd attach a clear tube to the actual stem, clear tube, and then fix that to the wall. And then make sure we wrap this really well, and the water would continue to rise up that tube over a matter of only a couple of hours. I can't remember the exact measurements now, but the water would go up to a height of about roughly 2 meters, which illustrates very, very nicely that concept of root pressure. So obviously there's no negative pressure because there's no shoot system, there's no transpiration occurring whatsoever, but water will still move up into the actual roots and up through that tube. Just roots, that's all that we had, just roots and a small part of the stem. That's all. We cut the whole plant off right here so that we only had maybe maybe about 10 centimetres of the actual stem. That's all that we had left. Quite amazing, and I think really nice to show the concept of positive root pressure, pushing that water up through that clear plastic tube. Really nice. And that's stated here. If the stem is cut off just above the soil, the stump will exude sap from the xylem for many hours. But those pressures are not very great. They can only push water up so far. And here's some values here for positive pressures. These are obviously positive values, equating to roughly 0.05 to 0.2 megapascals. So when transpiration is low, transpiration rates are low or absent, then positive pressure builds up because roots, of course, continue to actively transport solutes in from the soil solution, and of course this is followed by water movement. All right, that's all I need to say on that. Let's move on. So we won't talk about that. And I'll just clear my drawing. Okay, bulk flow, pressure-driven bulk flow through the xylem. Here we go. All right, we're all familiar with the theory of water movement. Cohesion tension theory has been referred to as the cohesion-adhesion tension theory. Water at the top of a tree develops a large tension. We're going to look at why that develops. It's a negative pressure, negative hydrostatic pressure, and this tension pulls water up through the xylem. The theory requires the cohesive properties of water to sustain large tensions in the xylem. So who can tell me the basis of the cohesive properties of water? What's the basis of cohesion of water molecules? Correct. Hydrogen bonding, yes, describes what we mean by cohesion of water molecules to each other. The negative pressure actually develops in the leaf, develops at the surface of the cell walls within the leaf. And we'll look at that in just a minute. Water adheres to the cellulose microfibrils of the cell wall of those cells, the mesophyll cells in the leaf. Then as water evaporates, we end up with, once again, just like we described for water in the soil, curved air-water interfaces. And this is where that negative tension, that tension or negative pressure rather, is initiated. Right here in the cell walls of the cells in the leaves. So here we have an actual image, a photo. This is a photo of the outside of the cell wall, and you can see all these cellulose microfibrils. Now, if we enlarge that, or rather just use this schematic here, which represents only two cellulose microfibrils, at an angle to each other. And here we have the air spaces within the actual leaf. And as the water evaporates from the outside of the cell wall, as the water evaporates, once again, we get this curved air-water interface. As it evaporates a bit more, the curvature becomes greater, and then greater again. And the greater the curvature of that water as it's evaporating, the greater tension develops within that water, the greater the negative pressure. And you'd be amazed at how this translates into, here we go, pressure, hydrostatic pressure. Here's the radius of curvature in microns. Let's look at that again down here. When the radius of curvature equals 0.5, here it is here, the pressure, hydrostatic pressure, is negative 0.3 megapascals. Let's say more water evaporates from there into the air spaces within the leaf. Here the curvature becomes greater, radius of curvature 0.05, here it is here. Look at what that does to the pressure. It's even more negative now, -3. And again, greater curvature of that air-water interface, radius of curvature 0.01. Now we have an even more negative hydrostatic pressure, -15. Quite amazing how that tension actually develops here. So here we are inside the actual leaf. We've got our cells. These are most likely spongy mesophyll cells because there's lots of air spaces in between them. And here's the water, the very thin film of water that forms outside the actual cells, on the cell walls, wet surface of the cell walls. This is where the actual transpiration process begins. So this is where water is evaporating into the air spaces, but it's also, as we just described, where the actual tension develops, where that negative pressure develops, right here within the actual leaf itself. Oh, I should have said this is the region that we're looking at within the actual leaf, and sure enough, they are spongy mesophyll cells. How's everyone so far? Has anyone fallen asleep? I just saw a comment there. You could hear someone snoring. That's why you've all turned off your mics. How's everyone else? Oh, that's very good. I like that. No, not at all. Not at all. You guys could easily follow this and more, but I won't give you too much more. Just a little bit more. All right, we've talked about that. We've talked about cavitation, embolism. All right, so now water is pulled from the xylem into the cell walls of the mesophyll cells. Of course, that's what we've just seen, the development of that negative tension, and of course that will pull the water up. And from there, from the actual outside of the cells in the leaf, the water evaporates into the air spaces. From there, water vapour exits through stomatal pores. That's pretty straightforward. Thus, transpiration itself, or movement of water vapour out of the leaf air spaces, is driven by the concentration gradient of the water vapour. Yep. That's the major driving force behind transpiration, concentration gradient of the water vapour. We know that the waxy cuticle is a very good barrier to water loss from other cells. And of course, about 95% of water loss is through the stomata, but there is a little bit that is lost through the cuticle. Okay, we'll move on a little bit more. All right, so still on transpiration, loss of water from the leaves. Transpiration depends on a couple of different factors. Of course, the difference in water vapour concentration between the air spaces in the leaf and, of course, the air external to the leaf. And we can look at the water concentration differences here in this table. So the inner air spaces, and this is all at 25 degrees Celsius, the relative humidity is almost at saturation, 0.99. Almost saturated. Water vapour concentration is here. What about the water potential in megapascals? There it is. So that's a number given there, negative 1.38. Just inside the stomatal pore within the leaf. Okay, still almost at saturation, but a little bit lower than the inner air spaces, 0.97. But look at the difference that makes to the actual water potential itself. We've gone from negative 1.38 to negative 7.04. And then what about just outside the stomatal pore? Here we've dropped down to 0.47 relative humidity, and look at the water potential now. Look at that difference, negative 103. Quite amazing that small differences in humidity cause such a huge difference in water potential. All right, of course you don't need to remember that. This is just to illustrate a few points here. That's all I'll say there. Okay, resistance to diffusion of water vapour. And I think that you would be familiar, at least in some part, with leaf stomatal resistance. So this basically is affected by the aperture of the stomata. Obviously, when it's open, there's very low resistance to loss of water vapour. When it's closed, very high resistance to loss of water vapour. Who remembers the leaf boundary layer? So this is another important resistance point to diffusion or loss of water vapour. Resistance to transpiration. Who remembers the leaf boundary layer? The leaf boundary layer, a layer of unstirred air saturated with water vapour right next to the leaf surface, which you can imagine if the water vapour is diffusing out here through the stomatal pores, that water vapour will accumulate just outside the surface of the leaf here, and this is what we refer to as the air boundary layer. The thicker that air boundary layer, then the greater resistance there is to loss of more water vapour from the actual leaf. And this can be influenced by the presence of leaf hairs, size and shape of the leaves, and the leaf orientation relative to wind direction. All right, that brings us to stomata. So where do we find stomata? High wind causes stomata to close. That's a very good point. Can anyone explain why high wind, particularly with high temperatures, lots of wind, high temperatures, why would that cause stomata to close? To reduce water loss. Absolutely, yes. Yep. So higher wind speed, high temperatures, the air boundary layer will become very, very thin or even virtually non-existent. So that basically increases the magnitude of that concentration gradient and increases the amount of water loss. And of course, if there's abundant water, then it's not a problem. But if there's any sort of deficit in water, then absolutely, stomata will start to close. All right, and actually you'll be doing a very, very nice prac during your prac workshop where we actually look at stomata opening and closing. So yeah, I'm looking forward to that particular prac. Particularly in response to application of a particular plant hormone. Very interesting prac. Okay, stomata. Let's look firstly, just very briefly, at the two major shapes that we encounter. Two main types. Firstly, we have the dumbbell-shaped stomata, and they're associated with subsidiary cells. These are found in grasses, and I'll show you what they look like here. There we have dumbbell-shaped stomata, or rather the guard cells have these dumbbell shapes, and here are the subsidiary cells on either side. And then in dicots and non-grass monocots, the guard cells tend to be kidney-shaped. And here's the kidney-shaped guard cells here. Two major types. Now, can anyone tell me where would you find stomata? Where would stomata be more abundant on the leaf? Upper side, lower side, both sides? What do you think? Have you read anything about this? Where is stomata found? Where are they most abundant? Underside? Yep. More abundant underneath. Yeah. Well, actually they are, but this also depends on the plant species. So certainly with dicots, they seem to be more abundant on the underside of the leaf. That's not to say there are none on the top side of the leaf, but there are plant species. So for example, the grasses, some of the grasses tend to have an equal number of stomata on both sides. Okay, so nothing additional there. Okay, because they're trying to dry themselves out. Oh, the grasses are. Yeah, I'm not sure about that. Actually, why grasses, or at least some of the grasses, tend to have equal number of stomata on both sides. Yeah. I'm not sure. I really can't comment on that. I've never actually worked with grasses before. All right, so let's go to your stomata lecture. All right, so we've looked at the soil solution, and here we have water movement up through the actual xylem. So looking at the actual stem or trunk. Now we've talked about cohesive properties of water by hydrogen bonding. What about adhesive properties of water? What's that referring to? So in your reading, what's this referring to? Adhesive properties of water molecules. Water tension? Not quite water tension itself. Although this would certainly contribute to movement, makes it adhere. What does it adhere to? It's what water sticks to, basically. Yes, we can say that. Maybe the curvature, adhesion. Yes. So basically what it sticks to. So let me just get the pen up. So everyone's familiar with, you would have seen this in the lab, certainly. If you have water in a test tube or in one of your burettes, for example, you would have noticed this. So this is the level of the water. Yep. Does everyone recall seeing, this is the water here. Does everyone recall seeing this in any of your lab classes? That curvature. Yes? Everyone remember seeing that? Do you remember what we refer to, what we refer to this as? Okay, not that it matters. I was just wondering if you remembered. No, the meniscus. Does everyone remember that, the meniscus? All right, it doesn't matter. It doesn't matter. So this curvature here, this curvature here, basically illustrates that property of adhesion, because the water molecules here on the side here are sticking to the glass. And the reason they do that is, one, yes, as a function of surface tension, which someone mentioned, surface tension, not so much water tension itself, but I think maybe that's what you're referring to, but also because glass and the inner walls of the xylem are hydrophilic. They're hydrophilic, and water molecules will attach or adhere to a hydrophilic surface. So that's what we mean here when we say adhesion. Water is actually sticking to the inner walls of the xylem vessels. They're joining together, staying joined together as a result of the hydrogen bonding. And there's one more property that's not described here, I don't believe. What about capillary action? Capillarity. Everyone's familiar with capillary action? Yes or no? I'm sure you are. Capillary action, yes. So if you have a dish or a container of water, there's the surface of the water, and then you put a fine tube in that water, fine capillary tube in the water. What will the water do? Yep, it'll actually move up that capillary tube so that its upper level is higher than the level of water in the actual dish itself. Yep. And of course, this is driven once again by the adhesive properties of water clinging to the inner walls of the capillary tube, the surface tension of water, absolutely. If we had another tube, another capillary tube, there we go, and instead of water we had mercury in the dish, instead of what we're seeing there, that curve there, we would have something like that. The curve would be that way because the mercury does not adhere to the actual inner walls of that tube there. Okay, so capillary action is also part of that cohesion tension theory of water movement. All right, so everyone's happy with that, and then of course we've moved up to the actual leaf itself, and we've looked at how the negative tension, the negative hydrostatic pressure, originates from within the leaf itself, due to those curved air-water interfaces. All right, we're nearly done. And we're just going to have a look at stomata. And I think that will actually complete tonight's class. Stomata opening and closing. What drives the opening of stomatal guard cells? So just looking at this image to the left. Pressure, yes. What initiates that pressure? What starts the process? Okay, without worrying about the actual stimuli that causes this process. Don't worry about the actual stimulus itself. Let's look at the chemistry of what's happening. Okay, so whether it's in response to light, and so on, what is the first part of this process? Which even comes before the actual pressure. What's the first part of the process here that we can see? Okay, ATP, all right, yes. ATP is used to do what? It's used to drive something. You're absolutely on the right track. ATP provides the energy exactly to pump protons out of the actual guard cells. Protons are actively pumped out of the guard cells. So this is a proton ATPase, active pumping of protons outside. What does this do? What does this result in? So if you're pumping positive ions outside, it starts the transfer, yes, yep, it certainly does. Active pumping of protons to the outside of the membrane actually sets up something across the membrane, sets up something. Which is referred to here as membrane hyperpolarization. Don't worry about that. What is it setting up? That's just a more complicated way of saying that we've set up an electrochemical gradient across the actual membrane. As a result of setting up that electrochemical gradient across the membrane, then this will trigger, starts the transfer of potassium. So we've got a potassium channel here. Potassium will move into the guard cells. This is something else that you'll be testing in one of your prac classes. Be looking for the presence of potassium in guard cells. Okay, inward movement of potassium. We also have inward movement of chloride. Yep. And that's driven by protons, yep, moving back in against their electrochemical gradient. Here we also see nitrate being moved in also with the aid of protons. But the most important thing here to note is that the solute concentration is increasing in the guard cell. Solute concentration is increasing. What's the story here? Malate anion. Malate anion, this is part of the breakdown of starch. And this anion is formed basically to help balance the inward movement of potassium cations. All right, but the most important point to note here is that the solute concentration increases, yep, which decreases the water potential, the guard cell water potential. So if we decrease the cell water potential, this results in, here it is, inward movement of water. And water is just moving in by osmosis through these aquaporins, so moving in along concentration gradient or a water potential gradient, and the water is coming from the surrounding cells. Then, as someone mentioned earlier, the pressure, of course, increases, cell turgor increases, and this results in stomatal opening, because turgor increase means that these cells actually move outwards, and that outward movement basically opens this pore. Okay, and then of course the opposite occurs here, and this results in loss of turgor and closing of the pore. All right, so I just wanted to revise that a little bit, and we'll definitely come back to this much later on, especially in preparation for prac classes. So that concludes our class for tonight. How's everyone feeling? Thank you, everyone, for your contribution. Before you go, would you like, feeling okay? Good. Would you like, so you know that these classes are only scheduled for every second week. Are you okay with that? Or would you prefer maybe another class next week? Or would you like to just keep them every second week? Which means that we won't have a class next week and we will the week after. Okay, so you're good? Yes? Amelia, are you happy with fortnightly classes? Oh, you would like weekly? You get lazy. Look, I'm happy to run a class next week, if you like. Leave it. Okay, so we've got 2 fortnightly and 1 weekly. The vote rests with you, Amelia. Okay, all right, we'll stick to 4. I think I can quite easily cover the major concepts in one class from every 2 weeks of lectures, as we've done tonight. So I've basically covered the major concepts from 3 lectures tonight. So if that changes as we go on, I'm certainly happy to run extra classes, but I'll leave that up to you. Okay. So thank you everyone for your input tonight. That was terrific. No, thank you. And I'll see you in a couple of weeks. But of course, let me know. Email Nikki or myself if you have questions about anything at all. Okay. All right. Good night.